Hey everyone, this is Miss Moriarty, and I'm here to discuss with us the end of concept number 4. So, the end of our discussion on cell transport and how the cell is going to transport materials in and out. So, remember in our discussions in class, we've been talking a lot about diffusion, specifically the diffusion of water, which we know is osmosis. We know that when looking at osmosis, a cell can be placed in 3 different types of environments that are going to influence how water is going to move. Now, each of those environments may be optimal for an animal cell or may be optimal for a plant cell. So, in this image, it kind of shows us what actually happens to each type of cell in each type of environment. So here we can see in a hypertonic solution, this is going to be a solution that has more solutes on the outside of the cell and less water. And that's going to force the water out of the cell, causing both cells to actually shrivel and shrink in size. We can see in an isotonic solution, this is where solute and solvent concentrations are the same. And so water actually enters and leaves the cell at the same exact time. So we see no change in either our animal cell or plant cell. Remember, we also said in class the ideal solution for an animal cell would be an isotonic solution because here it's at equilibrium. But in a plant cell, it's not necessarily the best solution. Now, looking at a hypotonic solution, we can see that this is where there's more water and less solvent on the outside of the cell, forcing water into the cell. Now, in an animal cell, because we don't have cell walls, our cells will burst. Versus a plant cell will become what's called turgid, right? We said in class that all that water is going to enter into the plant's vacuole, and that vacuole is actually going to swell in size, and that's going to create pressure. And that then turgid pressure is what's going to make the plant stand upright against gravity. So remember, overall, the ideal solution for an animal cell is isotonic, while the ideal solution for a plant cell is hypotonic. Now, the last piece of concept 4 is going to be discussing active transport. Right, because now we've just discussed passive, right? Remember, these are materials that move from a high to a low concentration without the use of any energy. And remember, we said that is simple diffusion, facilitated, and osmosis. But in active transport, it's just the opposite. Now materials go against the concentration gradient. So now they move from low to high. Now, in order to do that, that's a lot more difficult because things don't just naturally move from low to high. And so this requires energy. We can also see active transport when the cell wants to move very, very large molecules that can't even fit through a transport protein. We can see active transport being involved in that process as well. 3 examples that we need to know is 1, endocytosis. And actually, before we even go over exactly what this is, remember, we've seen this prefix endo before when we talked about the endosymbiosis theory. And if you can recall what that means, remember that was the theory that helped scientists prove how we got mitochondria and chloroplast. The fact that the mitochondria and chloroplasts are very closely related to prokaryotes tells us that, well, they were most likely once little bacteria millions and millions of years ago that were swallowed by a larger bacteria cell. And together they formed a symbiotic relationship where the little bacteria provided energy and the large bacteria provided protection. And through evolution, we then got the mitochondria and chloroplast. So in endocytosis and endosymbiosis, we saw that those then cells took things into the cell. So the prefix endo actually means in. While exocytosis, if you guessed it, is just the opposite. Exo kind of sounds like the word exit. And so this is when the cell actually pushes things out. And the last one is called the sodium potassium pump. This is a specific protein that's going to pump both sodium and potassium ions against their gradient from low to high. And so here, right, in this bullet, molecules that are actively transported against the gradient is our sodium-potassium pump. Things that are actively transported because they're just way too large to move in and out of the cell is endo and exocytosis. Now, specifically in endocytosis, we said this is where the cell is going to take large materials in. They can take in solid particles via phagocytosis. They can take in liquids through pinocytosis. And here they can take in signals through receptor-mediated endocytosis. So remember, in endocytosis, this is a form of active transport in where this cell is actually going to use its cell membrane to engulf something in the outside environment. Exocytosis is going to be just the opposite. So here we see that it actually expels unwanted particles or waste out of the cell. And you can see it's going to do that as a result of a vesicle. Remember, we said vesicles are organelles that are going to help transport materials around the cell. You can see that the vesicle coming from the Golgi apparatus here actually fuses with the cell membrane and pushes the waste out of the cell. And so overall, remember, endo is going to mean in, exo means out, the cyto suffix means cell. So when you put that all together, endocytosis means materials into the cell, while exocytosis means materials out of the cell. All right, guys, that's it for our video. Make sure as you watched, you took notes, and please mark this as done.